Hi, I am here now at Bio International with Kevin Curran, and he is with Biotech Primer. So, as you may know, we've been listening to these breakout sessions of our AiArthritis Voices 360 talk show. I am here, in addition with the organization, as a media person, and as such, the people at Bio said, "Who do you want to talk to?" And basically, I gave him a list. And that's where how this conversation came to be. So Kevin was doing a session on introduction to gene therapy. And, well, the introduction caught my eye. And it was, I think it literally said for non-medical.
Non-scientists.
Non-scientists. So I said, that's for me. So I'm going to let Kevin do a little introduction for himself and then also a little bit about your company.
Yeah, wonderful. Well, thanks, Tiffany. So myself, I'm a research biologist, so I'm a PhD in stem cell biology, taught at University of San Diego for a while, and now I am an advisor, consult in the cell and gene therapy space, but I've been teaching these non-scientist courses, teaching biotech for non-scientists in the industry for about 7 years. So Biotech Primer delivers these live classes to biotech pharma audiences that want to know more about the functioning and the science behind their drugs, and they especially want to know about the next 10 years. What are the new drugs that are emerging? So that's my specialty. We did a 1-day course on Sunday, and I did an hour course on gene therapy. And the funny thing is, most people connected to healthcare and even in the pharma industry are non-scientists and don't have a great science background. So most folks do benefit from a little bit of background biology. So that's an overview on our courses, and we can talk about gene therapy specifically.
Yeah, I think that just a little bit of an overview, because patients are starting to just learn, like you just said, and precision medicine is in the cancer space right now, moving into other diseases. And the research that we've been seeing in the rheumatology community is very strong on biomarkers and doing different types of clinical testing and different modalities in order to determine, well, prediction, prevention, diagnosis, and then what's going to work best in each individual person. So what does that look like? We could say precision medicine, but where I was really drawn to what you were talking about is it's a modality. It's one type of way to look at it. There could be at-home tests where you do a prick and you send your blood in. This is something on the other spectrum. So could you just talk a little bit about just an overview of what you kind of went over yesterday? Not the whole hour, but you know what I mean.
And I'm building off of what you just said.
Mm-hmm.
You can think of the advancement of medicine, right, as kind of a slowly turning wheel that is kind of kicking off good functional medicine that reduces suffering, hopefully saves people's lives, and if not, makes them more enjoyable lives. And you can think of it as kind of a two-part deal. One part is the technology of the medicine itself, which is always improving. And that's the piece that I spoke to, where we now have these new categories or modalities of drugs like gene therapy, the ability to insert a gene into a patient's body in a way that hits the target cells where that therapeutic protein is necessary. That's a whole new piece of technology. The use of viral vectors to deliver that therapeutic gene. That's just one example of a whole new field of technology that is now commercially available. But the other piece to keep that wheel moving forward is the accurate diagnostics and biomarkers and being able to know what it is you want to hit with the drug, right? Because it doesn't matter how good your delivery system is if you don't know what you want to deliver and if you don't know what cells to hit. So I think if I could broadly generalize, you have two extremes, right, in terms of disease indications. If you've got autoimmune disorder on one side and a genetic disorder, an inherited genetic disorder on the other end. The genetic disorder is very specific. It's very defined, right? A child is born They've inherited a mutation. It's one gene, and you can sequence that out and you say, okay, well, there's that mutation. And now we know how that manifests through the body to cause the symptoms. That's a really clearly defined target. It's a genetic target. And so you can create a diagnostic where you're just going to sequence out that gene, and then you see the sequence, okay, and then you think about what tool to use. Now back to the other end of the spectrum into really the less defined with, autoimmune and really any kind of immune-related disease. We can't track that back to one specific gene, right? If we could, we would call it a monogenic disorder. There's multitudes of genes, and we only understand a portion of how that contributes to, a hyperactive immune system or a underactive, underperforming immune system. And so you don't have those clearly defined genetic diagnostics, and you don't know what a good biomarker looks like. For example, like you can do a blood draw, but what are you looking for in the blood, right? With hemophilia, back to the clearly defined end of the spectrum, you're looking for clotting factor 8, a specific protein that's missing in hemophiliacs for hemophilia A. Okay, and so you know that the drug needs to somehow increase that level of that clotting factor protein. But when you start talking about, back to the immune diseases, what are you, if you do a blood draw, what protein are you looking for which subset of patients? If that's not established, you can't simply just choose the best tool for the job. Okay, let's go with RNA or cell therapy or gene therapy. You won't get such a tight correlation between treating the drug and seeing a response in a biomarker. So those are just broad strokes to explain why certain diseases are more challenging than others. Is because we haven't figured out all the biology or what we call the pathology of the disease.
Right. Yeah. So while we were, and I was taking furious notes like I always do in this, so I was actually following completely what you just said. So that must mean that you did a really good job yesterday because that part was covered. So the other component of why we're trying to teach patients is just really understanding that there's something also you talked about, and whether it's gene therapy specific or not, you were talking about how we don't know how long things will last. And could you just talk to that? Because that, I put like a real big asterisk on my notebook on that one because I never thought of it that way before. So
this is one of the big issues with gene therapy. There's been a huge push for cures and a lot of funding, a lot of, government initiatives. Everyone wants cures. We don't want treatments. We don't want to keep bringing patients into a hospital once a month for an infusion. People want a one and done approach, which makes sense. It's you want to push the button and all the bad things go away. That's the promise of gene therapy. And honestly, things are looking good, but the question remains, how durable, how long will the benefit last of gene therapy? And we can't really jump ahead to the finish line because it's so new. We just don't know. We've got Luxturna on the market in the U.S. for inherited blindness, and we've got Zolgensma on the market for spinal muscular atrophy. And we're still in that kind of under that 10-year level in terms of collecting a lot of patient data, and so we can track this. We can see how much therapeutic protein is being produced off of the gene that was inserted into the target cells. That's a very kind of molecular way to look at the durability. But then, of course, patients don't care about that. What patients care about is their clinical condition and their life and how improved their life is. If they're blind, can they see now? What's their vision? How strong is their vision? Is it reducing over time? And honestly, I don't think it's going to be a slam dunk. So, we are seeing that to some degree. There's some fall off with the Luxturna, and we'll see with the others. But it's an important question because if we're talking about, 10 years of benefit and then a fade out, okay, that's 10 years is good. And if that means every 10 years we need to redose, then that's okay. but to the point I brought up yesterday is if you use an AAV virus, then you might introduce immune rejection the second time you put that virus into your body. So the AAVs is the most commonly used tool to deliver the gene into place. So that's something that has to be worked out, especially because pharma companies are charging a lot of money in the 1 million plus area. So if it's not a lifetime of benefit, then that has to be worked out. Maybe the price has to come down. So this is stuff that's going to play out in the next couple decades, and honestly, this is just, we're all in this together. Honestly, everybody wants one and done. Everybody wants to go after these 7,000 genetic disorders and then move into some autoimmune. People are fighting tooth and nail to try to come up with the best gene therapy approach or cell therapy approach. So there's a lot of companies going after diseases, but right now, a lot of them are going after the same diseases. Kind of, you could call it the low-hanging fruits, where really clearly defined diseases with well-established biomarkers, right? Because, when the FDA is going to think about approving a drug, they want to see that the biomarkers are a good readout of the disease condition, clinical condition, because then they have a quantitative metric for how well the drug works. Right. So a clearly defined biomarker is one of the key criterias that will bring in a lot of pharma competition, because they'll see it as like a gettable target, a gettable drug. Like they have a higher chance of success because there'll be firm goalposts, right?
Right.
Yeah, and so if there's a lot of variability in the patient population already, then there's going to be variability in their response to the drug, right? Mm-hmm. So I'm just thinking out loud here, but with something with like a less kind of biologically defined group of diseases like autoimmune, it would probably benefit the whole community to figure out what are the meaningful subsets within there that we can define based on something quantitative, whether it's biomarker, genetics, proteins in the blood, or whatnot. Somehow to kind of segment that up into kind of a tangible targets to go after for drugs. And that would feed into the clinical trials, right? Because you would design the clinical trials using that inclusion criteria.
And those are great thoughts, because it really molds in very nicely to what we're doing at AiArthritis, in that we have a new data bank that we're hoping that patients can get involved in with the concept, or the moving forward to understanding the subgroups and educating them along with being involved in the research and understanding, wow, we're seeing patterns now. Why does this group seem to be responding to this drug? And then what in their blood or what in their proteins, what is the link? And so we're really invested in working hard on that. And something else to point out that you said was the price. When we're thinking about, how long will this work, or what is the longevity, and something that costs an awful lot of money, now we're going into health technology assessments or value assessments and access, and how, so you said how long the life expectancy. So now we're talking about quality adjusted life year, which is a measurement through that we'll go into in a different episode. But the fact is, all of this matters. All of the stuff that we're doing right now is going to equate to the word access, getting it moved over to the clinical practice, getting it adapted, whether it's the FDA in the United States, EMA in Europe, or anywhere. And then globally, where's that piece of the pie for the money going to come from? If it costs a certain amount and only lasts 7 years, but 7 years of seeing is pretty high quality of life, right? So how do you measure that? And again, that's a whole other conversation. But no, this has been great. Is there anything else that you wanted to mention specifically about the company? I can mention a couple great things that I just learned. Yeah. Okay.
Yeah, no, I'll just say, check out biotechprimer.com, the website. We do all sorts of live classes, and we hit the industry from kind of all sides, as well as the regulatory side. But That's it. It's been fun talking to you about
All
this.
right. It's been great talking to you. Thank you.